led to a further experiment done by not Azarinsky and Kleitman, but the unfortunately named Dement and Kleitman. They predicted that if you wake people up during REM sleep and ask them if they're dreaming, as opposed to wake them up during non-REM sleep, during REM sleep, you will get dream reports, and during non-REM sleep, you will not. And that's exactly what they found. In their mid-1950s paper, they reported that approximately 90% of awakenings from REM sleep produce dream reports, and less than 10% of awakenings from non-REM sleep. And that's about as close to a perfect correlation as you could hope for, given the nature of the phenomenon we're talking about. Subjective first-person reports from waking people up, in the middle of the night, and dreams are, of course, infamously forgettable. So that was really a striking confirmation of the prediction that flowed from their hypothesis that REM sleep was the objective equivalent of the subjective state of dreaming. Now, this was great because now we had something objective. Remember, psychiatry was dominated by psychoanalysis. Psychoanalysis was based on this free association method and fundamentally on Freud's findings about dreams. So at last, we had the possibility to be able to get out of that corner. And now that we had this objective physiological phenomenon of REM sleep, we could get a proper handle on what really drives dreams. This was made much easier by virtue of the fact that it's not only human beings who have REM sleep, all mammals do. And why that was important is because the standard way of going about determining which part of the brain is generating a particular function is to lesion, that is to say, to experimentally damage the different parts of the brain and see which part, when damaged, leads to a loss of the function at issue. So a chap named Michel Jouvet led the charge using cats. What I have on the screen now is a human brain, but human brain is sort of shockingly similar in its anatomy to a cat brain. And perhaps why Jouvet focused on cats was because they sleep so much. And what he did was really quite horrible. He sliced through the cat brain, progressively making deeper and deeper cuts to see at what point do the poor cats stop displaying REM sleep. And the surprise in this instance was eventually Jouvet made a cut here, separating the brain stem from the whole of the forebrain. The forebrain, including the cortex, is the organ of the mind. And remember what Freud was saying, that dreams are driven by these deep-seated desires and wishes and wants and so on. These are clearly mental forces. But when Jouvet made the cut here, effectively disconnecting the organ of the mind from all that remained, the brain stem, which is a very primitive structure, which was considered to be entirely mindless, sort of responsible for orchestration of bodily functions and reflexes. REM sleep continued in these cats, even though the forebrain as a whole had been disconnected. And it was only when Jouvet cut right down here to the structure which is known as the pons, it was only when he reached that level that REM sleep stopped in these cats. And so that showed that whatever it is that drives dreams, it's occurring at this level. Jouvet then passed the baton on to his pupil, Alan Hobson, and Hobson was able to identify precisely what it is that's going on in the pons. When we go into REM sleep, there are a group of cells over here in a region called the mesopontine tegmentum. These cells release a neurotransmitter called acetylcholine, which activates the forebrain. And then when we go out of REM sleep, these cells stop firing, and another group of cells, two other nuclei here, which I don't need to go into the details of, they start firing. So when these ones are on, those ones are off. When these ones are on, those ones are off. So What is it that causes REM sleep, dreaming sleep? It's a simple switch, on, off, and it switches on and off every 90 minutes, automatically, in the mindless, reflexive part of the brain that we share with all vertebrates. There's fishes have the same structures down here as. And so Hobson wasted no time. We've now reached the 70s. Jouvet's findings were the mid-60s, Hobson's were the mid-70s. He wasted no time in pointing out that Freudian dream theory was clearly wrong. This part, the acetylcholine, there's nothing motivational about it. There's nothing emotional about it. There's nothing mental about it. It's just randomly firing neurons in the cortex. The cortex, being the organ of the mind, is now activated. And so you have this chaotic, meaningless, mindless mental activity, conscious experiences, thoughts, memories, and whatnot. Hobson said that all that the cortex can do is join these dots, sort of make some kind of episode out of this random firing. To quote him, he said, "It makes the best of a bad job." And that's the dream. So this was called the activation synthesis theory of dreams. These pontine cholinergic, the cells that release acetylcholine, they activate the forebrain, and the forebrain, poor thing, synthesizes all of these inherently meaningless images, experiences into a dream. I say inherently meaningless. It's really important to get this point, that the thing that causes the dream, says Hobson, is this automatic pre-programmed clockwork of the firing of these cholinergic cells, which randomly activates the cortex, and the cortex joins the dots. You can come and look at that just as with an ink blot after the fact. You can look at this random activity, just like with tea leaves. You can say, makes me think of this or that. It suggests the following to me, but that meaning is not inherent in the tea leaves. It's not really there. It's not in the ink blot. You project it onto this otherwise actually intrinsically meaningless, mindless data. So that was Hobson's theory, the activation synthesis theory, and he presented it at a meeting of the American Psychiatric Association in 1977. After his lecture, a vote was taken among the members, and the question that they were asked to vote upon was, in light of Alan Hobson's findings, is Freudian dream theory still scientifically tenable? And not surprisingly, the vote went against Freud in Hobson's favor to the tune of 2-1. The ratio was 2-1 in Hobson's favor. And this was a decisive moment in the demise of psychoanalysis as a credible scientific foundation for psychiatry. 10 years after that, well, a little bit less than 10 years after that, I started the research program that I'm now going to report to you. What I did, I am a neuropsychologist rather than a neurophysiologist. And so I was more interested in the cortex than the brainstem. And this was in the mid-1980s. And we knew that the cortex does something rather more complicated than synthesize. This idea that the cortex is activated randomly during REM sleep, and what it does is join the dots. That's reasonable enough, but the different parts of the cortex contain different kinds of dots and do slightly more finessed operations than synthesize. So I predicted that, for example, if you have damage in the visual cortex, then the visual content of your dreams will be distorted or perhaps obliterated. That if you have damage in language cortex, that you won't be able to speak in your dreams, just as you can't speak in waking life, and so on. These are sort of fairly obvious predictions. So I wasn't questioning Hobson's theory. We all knew that the problem of where and how dreams are generated, the causal mechanism of dreams was in the brainstem. It was the same thing as the mechanism of REM sleep. But I was interested in the contents, in what does the cortex actually do that unpacks this notion of synthesis. What I found was really mostly, well, not mostly, it was entirely, in fact, serendipitous. The only one of my predictions that I think is theoretically interesting that was confirmed, I predicted that the prefrontal cortex, which I'm indicating on the screen now, this area here, which is our evolutionary pride and joy, us human beings, we got lots of prefrontal cortex. It's responsible for, it's equivalent to this pre-conscious part of the mind that Freud spoke about, the sort of verbally mediated, logical, rational, goal-directed agency, the sort of head office of the mind. I hypothesized, since during dreams, we don't seem to be in charge of our mental processes. They don't seem particularly rational and goal-directed. We are kind of passive recipients of what happens to us during dreams. I predicted that damage to this part of the brain would have no effect on dreams. And I was right about that. If you take the dreams, these patients with severe damage to the prefrontal cortex, they are very impaired in their waking cognition, but their dreams are indistinguishable from yours and mine. Blind raters cannot find any difference. They cannot differentiate the dreams of patients with damage to the prefrontal cortex and the dreams of you and me, assuming that you, like me, do not have prefrontal cortical damage. Here's a sort of summary of what I found. What I've just told you about was this part here, this dark blue area on this diagram is the prefrontal cortex here. As I said, damage there has no effect on the content of dreams. Now I'll tell you about some, I'm not going to tell you about everything I found, we don't have time. I'll tell you about the two most interesting things. And these were, as I said a moment ago, entirely unexpected chance discoveries, just good fortune. Patients with damage here, which is the mesoportal tegmentum, in other words, patients with damage here, although they lost REM sleep, and in fact, we knew before my study that human beings with damage there, like cats and rats, REM sleep is obliterated with damage there. I was not studying rats and cats, I was studying human beings, so I could talk to them. I studied a large group, roughly 350 patients, so it took me several years to collect this data. I had 18 patients here, all of whom, like previous research had shown, these patients do not have REM sleep, and yet my patients reported dreams because I was using subjective methods, in other words, talking to people about their experience. I was absolutely shocked to find all these years after the discovery of REM sleep and its correlation with dreaming in the mid-1950s. This correlation was, taken, the fact that REM sleep and dreams occur together at the same time, this correlation was taken to be an identity. It was taken to mean that dreams and REM sleep are the same thing. As I said to you earlier, REM sleep was taken to be the objective manifestation of the subjective process called dreams, and we then dropped the dreams like slippery fish because they're an embarrassment to science. You don't want to study this fluffy subjective stuff. So we then shifted to just studying the objective brain processes in order to get a proper scientific grip on what are the causal mechanisms of dreams. And so to my amazement, when I discovered that my 18 patients that I actually spoke to and gathered subjective reports from, reported, all of them, that they were still dreaming, I went back to the literature over the last 40 years and found that nobody had ever done that. Nobody had ever, although we knew that human beings, like other mammals, when they have damage here, their REM sleep stops.